So we're setting up our transformation lab today. We won't have results today. That'll be tomorrow. But we are going to do the actual part where we put the plasmid in the E. coli. Okay, 2 starts. We have these 4 lab groups set up. And to start off, we have 4 different plasmids here. Now I'm going to give you options. Okay, so option 1 is P green. This is the plasmid that has the green fluorescent protein and the ampicillin resistant, that antibiotic resistant protein. So it's that plasmid. Okay, we have P vib, which is this mystery plasmid that I found in the fridge. I have no idea how old it is, but it had to be in the last 3 years because that's the only years I've been doing this lab. So mystery plasmid that could be old, so may not work, but who knows what it is? Mystery. That's an option. Then we have PCAN, which is a plasmid with just a kanamycin-resistant gene. So it's a different antibiotic resistance gene. Only that gene is in this plasmid. Then we have our last one, which is PAMP, which is just an ampicillin-resistant gene in this plasmid. So talk with your group and figure out which plasmid you guys would want to use. We can't have multiple groups use one plasmid. Okay, so you can choose your adventure. Can. Multiple groups can use the same one. So
can you explain them again? There's the
P green is the green fluorescent protein plus the ampicillin resistant gene. The mystery one, I think, is the same thing as the other one, I think, but I'm not sure, so I can't guarantee. And then this one, the kanamycin, one has just a kanamycin resistant gene, one has just an ampicillin resistant gene. I think the mystery one is the same as the P green, which is the green fluorescent protein, but again, not sure. Found it in the fridge. Okay, decide amongst yourselves.
Yes.
Hot water.
All right, so here, let's discuss. So since some of you have chosen some different ones, I'll have to redraw one of these. But
I like our brand.
if you chose the mystery, which is P vib, is the mystery. And then we have P green. I think these are the same thing. So this is what I think is in the mystery one, but here's what I know is in the P green plasmid. You have your small circular piece of DNA. Remember that I'll just start all the way over.
Please.
So remember you had your plasmid, right? We were making plasmid hats. And the first thing you did with the plasmid hats was you made just a circular piece of DNA. You just made it into a circle. What's the first thing we had to do to the plasmid?
Cut it.
Cut it with a what?
Scissors.
Well, you literally cut it with the scissors, but it was representing what?
An enzyme.
What kind of enzyme? Restriction enzyme, yes. So it was representing a restriction enzyme. So the first thing we did was cut it open. Well, then what's the next thing we needed to do?
Put in the stuff.
What's the stuff? Cut the sequence out that you want of the genome, yes. And what was the gene that we put into our plasmid?
Insulin.
Yeah, we use insulin as our example because we're pretending to be a lab that was making insulin. Okay? The one that we're trying to get in with the P green plasmid, the one that we're interested in getting into the plasmid is that what's called the GFP gene, which is green fluorescent protein. That's what GFP stands for. So that's the gene from the jellyfish that will turn green and then will glow under UV light if it works correctly. Okay, so that's the gene that we want in that plasmid. And then we talked about the fact that we need to be able to select that plasmid out. So what do we have to add in here if we want to select only the bacteria that have that plasmid?
Antibiotic resistance.
Yeah, so that's why you put in that antibiotic resistance gene. So we had to cut it open again, use another restriction enzyme to cut it open again, and this time added another sequence that was ampicillin resistance. Okay, so we'll just call that AMP. Okay, so that is what I believe this one is, and that's what P green is. But you guys want to do the plasmid with the ampicillin resistance gene. So to make this plasmid, they just did the same thing by cutting open with the restriction enzyme. And the gene they inserted in was just the AMP, which would be the same as that. So in that plasmid that you guys have, it has only the ampicillin resistance gene. Okay? So I want you, let's all start, and I want to come back here after we do this to explain a few more things. But let's start by getting some supplies from the back. Send someone from your group to get one of these white trays that are filled with plates and other stuff, and grab, we don't need the pipettes quite yet, so no pipettes. So just grab one of the plates of stuff.
I think that's even the most hard job.
Because you're gay.
You're like, nose goes, you have to go get a tray. Okay, go get it.
Lance, will you go fill this with ice? Let me dump this out first. Who thinks so?
I like this.
Can we drink this?
Yes.
Or not?
I think you already knew the.
No, but like,
It's too cold.
are we
Yeah,
able?
it'll burn your teeth.
Well, E. coli was in there, so no.
Oh, that too.
But
So do you want to drink soup water?
So athletic
is this
training
a
room,
bar?
you know the deal.
why is it like to the top? All at the top?
All the way to top, yes, please. All right, so everyone's got a tray now, right?
Yeah.
You all have gloves on, correct? So you can touch the stuff in it. Good. Okay, so you have plates in there, as you can see. So when we're completely done with the transformation part of it, which is what we're going to be doing today, the last step you're going to do is plate out your bacteria so that we can see if the transformation worked tomorrow. So we're going to grow it on agar. That agar that you see is that like brown material. It's basically, you think of it like Jell-O, but with broth nutrients for the bacteria. And there are 3 different plates that you have. You have LB, so find your LB plates right now, the ones that are just labeled LB with nothing else on it but LB. Don't open the plates, but just find it. You should have 2 LB plates. Thumbs up if you have 2 LB plates. Thumbs up. Okay, great. So LB just means that it's just that nutrient broth. So all bacteria should be able to grow on this nutrient broth. The second set of plates you have are LB amp. That means that it's that same broth, but after I melted the broth down before I poured the plates, I added a vial of ampicillin to it. So there's antibiotics in that agar. The antibiotic that's in it is ampicillin specifically. Okay, so you should have 2 LB amp plates, and thumbs up if you have 2 LB amp plates. Yes? Yes? Okay, good. Okay, so then you have our last set is LB Canna plates. So with this is again the same broth, but this time we have a different antibiotic called kanamycin. So same procedure, just instead of ampicillin, I added kanamycin and then poured the plates out. So there's antibiotics in your kanamycin plates. So thumbs up if you have 2 LB Canna. Okay, great. So let's talk about what we should expect on these plates. So we're going to, with the E. coli, to have the plasmid, okay, where we expose them to the plasmid. All of your plasmids should have an ampicillin resistance gene, right? So if that E. coli, for the E. coli that does take up the plasmid, will they be able to grow on this plate, the LB amp plate, if they have the plasmid?
Yeah.
Yes. Why?
Because it has the antibiotic resistant genes.
because it has this gene in there that helps it to grow even when it's on an antibiotic plate. So only the ones that have the plasmid will be able to grow on here, because no other bacteria should be able to grow on the plate that has the ampicillin. Thumbs up if that makes sense. Okay, so if for the E. coli that take up the plasmids, will they be able to grow on the Albicana plates?
Yes.
Think about it for a second. Think about the genes they have if they take up the plasmid. Will they be able to grow on the Albicana plates? Okay, what do you guys think?
Yeah.
What?
Think yeah?
Yeah.
What gene do they have to be resistant to? Do they have a gene that helps them be resistant against kanamycin? So what gene do they have to be resistant against? Just AMP. So do you think they'd be able to grow on another plate with a different antibiotic? Most likely not. Okay? Because they don't have a kanamycin resistant gene, they have an ampicillin resistant gene. There's two different methods of action for those antibiotics. That's why if you're sick with something, and say, again, we'll just use strep throat because that's what a lot of people get antibiotics for. So you get strep throat, and they're like, okay, here, take this. I think this is what you're infected with. Take these antibiotics. And what if it doesn't work? They go, oh, crap, what's going on? So you go back. They'll say, oh, gosh, okay, this did not work for this particular bacteria. Let me give you this different antibiotic. And that one knocks it out. Why did the second one work? It wasn't resistant to that antibiotic, but it was resistant to the first antibiotic. Okay? It only will be resistant to the very particular antibiotic that it has a gene to be resistant against.
Isn't that like a big issue, like antibiotic resistance?
Yes.
Not yet.
Yes, it is. Yeah, there's a lot of, we're going to talk about antibiotic resistance and evolution, and we're going to have a whole lab just for antibiotic resistance. But the concept is we're going to take advantage of that concept here to select out these plasmids. Okay, so let's go now to the LB broth. So the plates with just the broth on there. Will the ones with the plasmid be able to grow here?
Yeah.
Yeah, it's just nutrient broth, right? Will all the other E. coli be able to grow here?
Yeah.
So everything can grow here.
Yeah.
Can everything grow here?
No.
No. What are the ones that can grow here? The ones that have the plasmid. Okay? What can grow here?
Ones that have the camera resistance to.
Do any of our plasmids have that?
No.
No, unless the mystery one, but I don't think it does. But having these two plasmids, no. So should any of our bacteria grow here? No. So you would be successful if you saw no growth here. Okay? Questions? Okay, now let's talk about how we're going to do this. Okay. So, there it is. So go ahead and turn your page to S7, and I am going to come and check your pre-lab questions, by the way. But that'll be during your lab.
It's unneeded.
So let's just double check that you have the materials you need first. You have this nice little graphic.
Got the scientist.
Yep.
Check.
They can check on that
We
one.
literally ran
We
out
got
of
a
time.
huge check on that.
Yeah.
We got your mama.
So we have, you have what plasmid you're going to use. You all have that, so check. We have all the plates. Check on that. Do you have 2 transformation tubes?
Yeah.
You should have 2 transformation tubes.
Yeah, we have those.
Great.
Yeah.
And you should have a tube rack. If you don't have a tube rack, there's one right there. So you guys, I think, just need to take that stuff.
Close enough.
Okay, so go ahead and put those tubes in that tube rack. That's what you're going to use to hold that.
Okay.
Do the honor.
Go ahead and label, we'll do the first step together. Label one of those tubes with your Sharpie. Label one tube plus plasmid and another tube minus plasmid. Doesn't matter where, just at the top somewhere. plus plasmid and minus plasmid.
Okay.
Okay. We've all done that or are doing that. Great. You should have 3 sterile inoculating loops, which are those little yellow things in a package. They're sterile, so do not open them until you are ready to use them. Okay? Because they are clean, but if you open them, they will not be clean. Then you should have the transfer pipettes. I'll talk about in a second. We're not going to use the pipettes that they want us to use. You should have a waste container with bleach at the very bottom where you have some used stuff from the other class where you put all their pipette tips in there. Yes, Rand. And then you should have a vial of LB, so that broth. And then you'll have, everyone have the broth?
What?
Yes, broth. No, you should have a vial of broth in your container, little tiny vial. It says LB.
This?
Yes.
Yeah.
Good. Okay, and then you have an ice water bath there, right? So I'm going to have you guys pour out the water in that. Pour out most of the water in the sink, either the sink next to you or the sink back there. Pour some of the water out, and I'm going to fill it up with ice, give you a refresher of ice. Don't pour all the water out, just the excess water. Have a little bit at the bottom. That will help your tube stand up.
Marshall.
Okay, shut up, stupid.
A little bit more than that.
Yeah, you put it on Marshall.
Dude, quit dumping the cubes.
Either you, need to,
Okay, that's good.
Come on, Marshall. You dumped out enough.
Hey, Lance, I think we're going to need a refresher.
Lance says refresh.
She likes to say refresh.
Do I?
Yeah, you said it 1,000 times, I swear.
He swears.
I swear.
All right, I'm going to give you a vial of calcium chloride that is ice cold.
Or the baby's gonna beat you up. AKA me. I am.
He says he's.
I'm the baby.
I said on the baby.
That's more like.
Lance, we may not need a refresher.
See, I'm
Who's Matthew? I know you're.
Here? Or
Matthew's
you?
like a little kind of low key.
Or you?
Matthew's a pretty shit.
Okay, I'm so sorry.
Well, what?
Okay, he's like young Greg. Really, obviously.
Happens on November.
And I'm like little
Put
Uzi,
ice
so
in
we're
here.
like the same height.
A little bit.
You know?
Lance, we really need a refresher.
What's his name?
I was looking for
Machine Gun. Machine Gun Kelly, MPK. Yeah, that's you. All right.
Thank you, Lance.
I'm
I
like,
see myself more as like a clean M&M type rapper.
Clean M&Ms, that's-
Yeah, exactly.
you mean that's
There you go.
standard. That's standard.
Hey,
What?
take it.
Why? Wait, what is this?
Clean M&M.
You'll need it.
What am I supposed to do with it?
Genghis Khan
Put it on your
comes back
table.
alive.
This is why I don't give them the laptop.
Yeah. Bring him back. It's like.
Okay. So we have all these things now. Great. Lance is going to refresh your ice bath so that you have a perfect ice bath. All right. So when it says you need to use a water bath, because you each have your own ice bath you'll use, but then it says a warm or hot water bath, and that's back here. So when you need to use the water bath, you're just going to have someone from your group. It's only 90 seconds that you need to use it for. Just sit in there and hold the liquid in there for 90 seconds. Put it over. Okay?
Okay.
That's the warm water bath. And now let's go through I'm going to go through the more confusing steps at the very beginning with you. So turn to the next page. It says procedure.
Yes.
Why do you say bath?
That's what you call it, a water bath.
That's not
Okay,
That's
so we've already labeled our plasmid, so we've started off. Now it says to use your ice cold calcium chloride. And you're going to take 250 microliters and put it into both tubes, as it says there. But for pipettes, most of our pipettes will not go over 200 microliters. So I have 2 pipetters here. I have 2 pipetters back here that will do 250 microliters. They're the big dark blue ones back here. So I have 2 dark blue ones, and you're going to have to use the really big pipette tips for this. The big pipette tips are in this container, this bright blue container. You guys are just going to have to share these. There's 2, and you'll only need it for like a hot second. It's already set to 250, so you don't need to change it.
Don't change
So the first thing you're going to do,
It's at 25.
never mind, you don't get it. Just kidding. That's 250 on that one. That's why I set it for you. Okay, so you'll be using that to measure out 250. There will be a time where you need 100 microliters. You can use a green for 100 because it does 20 to 200 microliters. And you remember the pipette tips that you used for that was these.
Oh my
What
God,
is
we
that
can't try shooting.
Throw it on the ground. That was the best response. I know. And then you'll have one time you'll need 10 microliters, so then that's the yellow guy.
Where'd
Alright?
that pants go?
You don't need to come grab those until you actually need them. Thank you so
it.
much.
Good job, Lance.
Lance, will you fill those 2 just a little bit? There's like a handful of ice to each of theirs. Thank you.
You need a refresh.
Okay. So, in these first couple steps here, so I'm on, we just talked about number 2, okay? Number 3, it says you're going to put the tubes on ice. When you set them in your ice bath, make sure that they don't tip over and let water in there. We gave you enough ice so that they should be able to stand up on their own. But you're going to take those tubes one at a time, and you're going to add bacteria to both of those tubes. Okay? So you're going to add bacteria, you're going to use these inoculating loops. So I'm going to show you how to open these containers. You can see on your loops that you have a loop side that has like a circle, and then the other side you have a pointy end. Open it on the pointy end. Okay, and just tear it apart. Don't do it until you need it. I'm just doing it as an example. You don't need, it's sterile, so don't open it until you need it. But you're going to use the loop end on the actual auger. Lance, scoot forward for me. You're right in my way.
We have 3 of these bad boys.
Good.
We only needed 2.
Okay, so you're using the loops to get the E. coli from the auger. Just use a new one for each tube. Okay? A colony, it says to get 1 colony. 1 colony just means 1 dot. So you can see it looks like streaky on some parts. Don't touch that side. Only the colonies, the ones that are in circles. Because you don't want to use, if you use too much, then the reaction won't work if there's too many cells in there. So you only want to use 1 colony per tube. So if you see an isolated dot, you can just gently scrape on the auger. And this is Jell-O, remember, so you don't need to stab. It's just a very gentle scrape. You don't want the auger to come with it. Just gently slide it over and then put it in your tube and just shake it to try to get the colony out. It's going to look like you have nothing on your on here at the very end. But you will have bacteria on there. You're just not going to be able to see it because it's almost translucent. Okay? But you only need just that little bit. That is plenty. There's a lot of bacteria.
So, what are we supposed to be talking about?
So just 1 dot. See that like 1 dot right there?
Yeah.
You would just want 1 of those per tube. Okay? I can help you if you're here and you're confused, because I know it's hard to see from over there too. If you can't figure it out, it's better to ask me than have it just do what you want and then have it go wrong. So there's 4 plates over here that you will use for that. 4 different plates, so you each can have a different plate to take the bacteria from. The ones you're looking for are the ones with the crosses on the bottom. Okay, so that's where you're going to get the E. coli from for that next step. Then you're going to shake it in each of those tubes. Just do 1 tube at a time. And I think those are the most confusing steps. Oh, then you're going to put them on ice for You're going to put them on ice, and then you're going to add the DNA. Here's the other part. I want you to kind of scratch out number 9. Because I'm going to tell you, just scratch it out because I'm going to tell you what we're going to do. Because I don't like the way they have it here. So I'm changing it up. So you're not, it says add a, well, I just won't even go over what they do. Just cross it out. You guys are going to add 10 microliters here. So next to that step, I want you to put add 10 microliters of plasmid. Remember, that's the yellow pipetter. Make sure you're using the right pipette tip. So you're going to take the yellow pipetter to get 10 microliters of your plasmid. Your plasmid is the tube that you've been assigned to, that you guys picked. Okay? Get your 10 microliters out of here, and you're going to put it in only the plus plasmid tube. Okay? Only add the 10 microliters of plasmid to the plus plasmid tube. You are not going to add it to the minus plasmid tube. Okay? Plus P, that's fine. Okay, so when you're getting it out of your tube, make sure you actually get the liquid that's in the pipette in the bottom of the tube. You're going to have to stick it down in there a little bit. That's fine because we're going to sanitize them at the end. So if you have to stick it in the tube to get it all the way to the liquid at the bottom, totally fine. Does that make sense? Pipette, just like you guys did yesterday, you guys did beautifully. That's why we practiced before we got to today. Okay, then you're going to put it in a warm water, an ice bath for 15 minutes. Yes, you have to sit there for 15 minutes. Then we're going to switch to the warm water bath. Why are we going from the cold to the warm water bath? We're stressing out the bacteria, so they do what? Yep, they collect the DNA from their environment. Should the negative plasmid take up the DNA from their environment? No, because they don't have the plasmid, right? But yes, they'll be exposed in the same way. Do you guys have questions about anything that we're about to do? Any questions about anything right now? All right, go ahead and get started. You are on the calcium chloride step right now, okay? Because we already did step 1 together. So there you go. Step 2, here we go.
Go get the fan. Yeah.
250.
Give me.
Is that the?
Okay, we'll put it around them,
Give it to me.
not her. Okay, here's your lab group. Say hi, lab group.
Hi, lab.
That's
Hi.
they shall guide you through the lab. Just pause it anytime it's to the point where it's like, wait, 15 minutes.
First place champion.
Da, da, da.
Yeah, we're gonna do good at step 1.
Yeah, what do we have? First, second, third?
One of these guys gets kind of stuck ejecting the tips. I'll tell you how to do that.
No, you're first.
Suck it up there.
Because sometimes
It's gonna be a little bit.
Give me.
All right, so
Okay,
basically
I wanna do it.
we're taking
There will
250
be more than one
microliters
opportunity
of calcium
if I
chloride solution, and we're adding it into
Both tubes.
each tube. I knew that, Emma.
All right, Tanner, cut it.
Bert.